Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to talk about a little bit in details on homoviscous adaptation. So let's get started with the video guys. So firstly the lipid and protein composition, because in previous video we have already talked that our cell membrane is composed of lipid, protein and carbohydrate. So what happening? So this lipid and protein composition of biological membrane varies among organism, tissues, cells and intracellular organisms. Okay, so that is not the main thing. Along with that, the membrane lipid, which is amphipathic in molecule that we have already discussed, this membrane lipid can self-assemble into 4 type of supramolecular structure. For example, they may be exist in micelles, they may be exist in bilayer, they may be exist in hexagonal structure, or they may be exist in cubic phases. Okay, so that is not our topic of discussion. Our topic of discussion today is the lamellar bilayer or the normal bilayer that we have just talked about. Remember about the 4 supramolecular structure I have just talked about. Among them, 2 is very important. One is lamellar bilayer, along with that our mycelia structure. Okay, so this lamellar bilayer have several physiochemical properties. For example, phase behavior, difference in membrane fluidity and viscosity, then membrane rigidity, and as well as membrane thickness and bending rigidity. Okay, and this type of physiochemical properties are actually determined by their molecular composition. Very important. And this molecular composition meaning the composition of lipid and protein, especially lipid. Okay, along with that molecular composition also membrane curvature. So it is very clear to us that to determine what will going to be the physiochemical property of a lamellar bilayer, we have to first analyze the biochemical composition of the lipid and protein, which are their composition, right? So along with that lipid, these biological membrane proteins will also going to play a very important role in a cell membrane, because they will going to introduce functionality in membrane. And this membrane protein can serve as a receptor, it may serve as a transporter, enzymes or structural elements. Let's consider a situation. If you may probably heard about the cell signaling, you may know about the cell signaling concept, then it is very clear to you that how much specific a signaling process is, right? The signaling molecule first have to find out its receptor that may be on the cell membrane or maybe inside the cell, but as here our topic of discussion is cell membrane, so we are considering that receptor is present on the cell membrane, right? So that particular signaling molecule have to recognize that receptor to facilitate further downstream signaling, right? And for recognizing the receptor, first you have to analyze what it have to analyze the composition of the membrane, right? So now let's talk about a situation where a cell signaling process, a particular cell signaling process is not necessary for the cell, or it may be detrimental for the cell on the basis of that particular environment where it present. Now what a cell can do to prevent that cell signaling, it may change the membrane composition so that the signaling molecule when it comes cannot find out its receptors, right? And other, it may also block the cell signaling during other downstream processing also, but it may also do the thing that we have discussed, right? So for changing this, it first have to know what is the membrane composition, or what is the membrane composition, or what is the lipid composition, right? So in this video we will try to find out what is the properties of lipid and what kind of function a cell membrane has due to different chemical composition of the lipid. Okay, so that is the idea. So as here I have told you that therefore a cell must monitor the membrane properties to mount adaptive response as well as to maintain organelles identity. Because let's consider a situation, there is a cell signaling process which is very important for the cell during summer or when the environment is hot, right? But when the cell is in cold environment, does that particular cell need the same signaling process? No. And it may be also detrimental for the cell, right? So the cell can simply prevent it by changing the membrane composition, and for changing the membrane composition it is necessarily important to change the lipid composition, right? So here we will going to discuss how change in the membrane lipid composition change a membrane's property. Okay, so that is the idea. So lipid has a pivotal role in membrane remodeling process. I hope you have understand it very well with the help of the example that I have given. So their biosynthesis as well as turnover should be tightly regulated. Okay, so although now we have learned that the physiochemical property that the lipid bilayer have is tightly depending on what is its chemical composition, right? But it only give us idea about simple type of physiochemical composition, physiochemical properties, for example, viscosity. But it cannot tell us about the bulky physiochemical properties. And for understanding those kind of things, we have used several type of technologies, for example, mass spectrometry along with subcellular fractionation, also immunoisolation of organelles that have provided us the insight of lipid diversity and distribution. They have told us how much amount of lipid a eukaryotic cell and organism has and their diversities, and how a eukaryotic cell have a gradient of lipid depending upon their needs. So let's get started the discussion. So eukaryotic cell and organelles have 100 to 1000 lipid molecules, all differing in their molecular structure, and as they are differing in their molecular structure, obviously they should have different physiochemical properties and also molar substances. That is molar abundances. Sorry for that, molar, it is not molar substances, it is molar abundances. And this stunning diversity is actually due to the combinatorial complexity of the lipid building blocks. If you have watched the basic videos on lipid, then you may know about how a small change in a lipid can give you a completely different one, right? Because glycerophospholipids, sphingolipid have different type of lipids that have a small difference in their structure, right? So that is the idea, and this small difference in their structure will bring a different physiochemical properties. Okay, and that is why this kind of lipid will present in different type of membrane. Okay, that means the lipid which is present in the neuron should be different from the lipid which is present in the endoplasmic reticulum. Okay, so that is the idea. So glycerophospholipid and sphingolipid, we are taking an example that this glycerophospholipid and sphingolipid has in general two apolar hydrophobic fatty acyl chain and one hydrophilic head. Two fatty acyl chain also widely different when we talk about their subgroups. They differ in their, they may be a fatty acid, they may be fatty alcohol, or they may be long chain bases differing in their length as well as number and position of double bond and hydroxylation. Not only that, the hydrophilic head we have talked about have variety of chemical structure. Okay, so you may understand how much diverse lipid compositions are in eukaryotic cells as well as eukaryotic organelles, and that obviously brings different kind of physiochemical properties. For example, the degree of unsaturation determines the efficiency of lipid packing in the cell membrane as well as membrane viscosity and water permeability. Because you know what, the unsaturation means what? Unsaturation means presence of double bond in the fatty acyl chain, right? This presence of double bond in the fatty acyl chain introduce kink. Okay, and this kink creates space. Okay, that is why the higher the unsaturation, the higher will be the membrane fluidity. Okay, now this amount of unsaturation should be very well regulated, right? Because if unsaturation is higher than needed, then it may be detrimental for the cell, as well as if the amount of unsaturation is lower than needed, it will again detrimental for the cell. So cell have to sense the membrane composition for its own survival, right? So that is the thing. For example, poikilothermic organism, for example, bacteria, cyanobacteria, then fish, fungi, plants cannot control their body temperature, right? So they will do, when they are in the cold environment, they will increase the component, they will increase the amount of unsaturation in their fatty acyl chain of the cell membrane. Why is this so? To increase the membrane fluidity. And what will the reason behind it? Reason behind it is, if the amount of saturation will be higher in lipid, then the membrane will be tightly packed. Okay, and it will not good for the poikilothermic organism, especially in cold environment. Okay, so that is the idea. So you can understand that unsaturation in the fatty acid chain affects lipid packing, membrane viscosity, as well as water permeability. Now this is about the prokaryotic organism, but the temperature will not going to be enough to explain the amount of unsaturation in fatty acid chain in eukaryotic organism, because in case of eukaryotic organism the things are completely different and much more diverse. If you analyze the lipid composition of in eukaryotic organism, you will find that eukaryotic cell have gradient of lipid with sterol and saturated fatty acid in the secretory pathways, as well as neuronal cell, especially in the axon tip you will find polyunsaturated fatty acid chain, as well as you will find saturated fatty acid as well as polyunsaturated fatty acid in the apical plasma membrane of the epithelial cell. Okay, similarly the amount of unsaturation will differ depending upon where that particular lipid is present. Okay, now why is this so? Because this polyunsaturated lipid has various type of properties. For example, they will adopt different shape, they will adopt stabilize, they have the ability to stabilize coexisting membrane domain, as well as they have the ability to facilitate membrane deformation. And depending upon the need of that particular plasma membrane, the amount of unsaturation will be present in that particular cell. For example, in neuron, ultra-fast endocytosis is necessary, and that can only be done with the help of polyunsaturated fatty acids. Similarly, signal transduction by mechanosensitive channel as well as photoreceptor is very important, and it can be done when the membrane lipid have high amount of unsaturation. That is why the cell which requires photoreceptor signal transduction by membrane sensitive channel, in that type of cell you will find a increased amount of polyunsaturation, and where monounsaturated fatty acid will be necessary, in that type of cell you may find monounsaturated fatty acid. That is the idea. That is how, depending upon the function of the cell, the amount of unsaturation changes. For example, here you can see that sterol and saturated fatty acid chain exist in the secretory pathway because they will play important role in the secretory pathway. So those cells who are involved in this secretory pathway, there you may find the increased amount of sterol and saturated fatty acid. So that is the idea of this video, where we have discussed that the lipid composition in the membrane is not a static thing. It is very dynamic, and it is totally depending upon in what environment that particular cell is present and what the cell wants. Okay, if the cell rapidly involved in cell signaling, then the composition of the membrane lipid should be different. If the cell is in a polykilothermic organism, obviously the composition of the cell membrane differs depending upon where it is present, right? So cell membrane as well as membrane lipid is not a static thing, it is dynamic, and it will changing with the environment. That is the main motto of this video, to understand. Okay, so I hope this video will be helpful for you, although I observed that it is little bit boring. So thank you for listening to this class. Hope this video will be helpful for you for understanding the concept. So bye bye. We will going to meet again in the next video.